All right, let's continue with annotating our assemblies. Um, Basically, exploring our blast hits a little bit more, um, and basically attaching our blast hits to our FASTA file. So we have let's go take a look around. Um, okay. All right. So just to kind of familiarize ourselves with this, with what's in this directory, um, the newest file here is going to be the reference orfs. I'll get to that in a second. But right, we have the longest .pep, we have the longest CDFs, the GTF, and the longest orf.blast. That's the stuff that I made. Yep, Siren Wednesday, no doubt, of course. Let's see, so longest blast. So let's take a look at that blast output file. I'm just going to head it real quick. Okay, and let's see. So again, we've already seen some of this stuff already, right? So we have the query sequence, our symbol transcript, a hit for it. This is a percent identity, like the length of the hit. I think this is the number of mismatches. That's the number of gaps. And this is the start and stop of the query. And this is the start and stop of the hit. Then we have an E value and a bit score. Right. This is just the product of the out format 6. Again, there's almost a dozen different output formats that Blast can give you. It's just the tab delimited is the easiest to look at and work with. And so really the only columns that I care about in the default state, is the query, the hit, and the E value. Now, sometimes I do care about the bit score. If I'm doing stuff seriously, if I'm really kind of looking around and playing with my stuff, I'll pay attention to the bit score, but not so much for our purposes. But if we scroll through here, so let's see, the second transcript we have has several blast hits to it. Yeah, all of this stuff reflects a blast hit for this one specific transcript, this DN10043. And so there's probably even more of them. But if we even just look among these lines themselves, right, so our gene of interest, or at least our best hit, is this PCX2 for human. And again, all of this is just the annotated, this is just the format, this is like a header within the Swissprot. Right, so if we searched through the Swissprot, looked for headers, we're going to see effectively this header. There will be a little bit more information to it, but these are the important things. Like this Q number is a specific accession number for Swissprot that links to the ANKF1 human gene. But of course, everything has a homolog. Almost all homologs are present in multiple species. So, right, so our first, for this specific DN10043 transcript, PCX2 human is the best hit, but a close second is PCX2 from the mouse, right? No surprise. However, this gets a little bit tricky with Blast, and it's always a concern with Blast because sometimes the best hit will not be a homolog. It won't actually be an ortholog, it'll be a paralog, right? So a gene copy. And so PCX has several paralogs, right? So PCX1 is a different paralog. PCX3 is a different paralog. Right, so you see that within all of these hits, all of the hits are within the PCX family, but it turns out that the best hit given the e value here is this, that's the smallest e value among all of these hits, right? 8.34 times 10 to the negative 105. That's the best blast hit. Also, the bit score is also the lowest among all of these, or the biggest, right? Among all these hits, 370 is the biggest bit score. That corresponds to the smallest e value. So that's kind of why I'm ignoring the bit score a little bit, because most of the time the e score or the e value and the bit score are going to mirror each other, right? The lowest e value will have the highest bit score. All right, and so if we look through all of this, right, we're just going to see this over and over and over again. Actually, I'm curious how many of the 443s actually were annotated. So 1, 2, 3, 4, 5, 6, 7, 8, 9 different possible annotations of DN10043. But they all blast to the PCX family, whatever that family is. I don't know what that family actually is, but nonetheless. And so we'll see this over and over again. Like if we explore this output file, so I'm going to do some cutting and sorting on this output file to more or less look at how many annotations, how many sequences were actually annotated. So first, I'm going to count my sequences in longest ORF, just because I kind of forgot the CDS file. 181,000 transcripts were present in that. So if I cut on the first column, so the first column is just all of the hits that were found, or query sequences that had a hit. Longest.orf. I don't have to sort this; it's already sorted, so I can just do a unique in it, a unique -c. I don't want to do that. Unique WC. So 78,000, right? So 78,000 transcripts had a blast hit. So a little less than half of all the transcripts that we found an open reading frame of had a blast hit to the Swiss Prot database. So I'm not going to worry too much about this discrepancy. Right? In a perfect world, every open reading frame is going to come from a gene that we know about. But ORFs can be artificial, right? It can look like an open reading frame, doesn't necessarily mean it is an open reading frame. Just because it's a long string of codons without an interrupting stop codon doesn't necessarily mean it's actually an open reading frame. It could just be a long sequence of non-coding data, and there just happens to not be a stop codon anywhere in it. It can happen. Other times, like the ORF that we find is just too small, it's too short to find an appropriate hit for it, right? The blast search will fail, right? We set parameters to the blast search, and if it doesn't cross those parameters, it's going to fail. And still, we have a lot of sequence to work with. 78,000 transcripts is probably pretty close to what I would expect in a protein coding transcriptome. Given about 10,000 different genes, several different transcripts, possible transcripts per thing. Yeah, all right. And so, if we see how many actually unique genes were annotated, we can more or less do the same thing for. I don't want to do that. I don't want to do that yet. I don't want to do that yet. I got caught off guard. But yeah, so the next kind of step is to, or at least for us, is to attach the best blast hit to these query sequences. Right, and the best is usually the first in this file. It's usually outputted first, right? So if we list this file again, right, there's the DN443s. Here's a new transcript, DN4021. And so you should see more or less the same pattern. The first hit is going to have be the most similar. It should have the lowest e-value and the highest bit score, right? So if we find another transcript, that same pattern will be present. DN40043. First hit is the best one, highest percent similarity, lowest e-value, highest bit score. And so these are what I want to attach. I want to attach this information to this sequence, right? And I want it to be in a FASTA format because I want to be able to map reads to these transcripts, and I want to know what reads, what genes I'm actually mapping stuff to. And furthermore, since I know that half of these transcripts are either poorly annotated open reading frames, non-existent open reading frames, or just too short to annotate, I want to ignore those as well. So I really only care about the transcripts that have an appropriate annotation, that have a good blast hit to our Swiss prod. And so I made a script to do that. We're not necessarily going to go over that script, just how we can go get that script. So this script is currently hanging out on my GitHub. Right, so here's my GitHub. And this script, this new script that I just added, is annotate.orfs.py. It is a Python script, and there is a little bit of information about it, what it does, and how to use it. So what this does is, like, given what we've done up to this point, it's going to add the best blast hit to our sequences, to our query sequences, and output it output our CDS as a FASTA with the raw transcript information, but also the best blast hit. So you don't necessarily have that script in your homes yet, or in your directories yet. So how you can go get this is to go to your Bio 435 folder, wherever that's at. Mine's in software. I don't know where yours is at. Wherever yours is at. Right, remember we downloaded all this stuff from GitHub. Like all of these scripts that you have in Bio 435 were downloaded from GitHub. So how you can update your GitHub repository is doing just git pull, git-pull. Now, whenever you run this command, what's going to happen is it's basically going to update all of the changes that I've made in my GitHub, and it's going to make those changes to your directory. So it's going to add the annotate orfs, and it's going to change the README, right? Because your README won't have the information about running annotate orfs. But after you run git pull, you'll have both the script and you'll have an updated README. In which case, then you can run this script in your in our blast output file. So I'm going to go back over there. Okay, I'm going to remove reference orfs. Okay, so I'm sitting where you should be sitting at. So I'm going to call this program annotate orfs, and then I call the FASTA file first. So I'm going to call my longest CDS file first. So longest orf CDS, and then I'm going to call the blast output file. So I need both of these files to be provided. I need the sequence files in the form of the CDS, and I need the blast files, longest orf blast output file. And so I'm going to save that into reference orfs reference .fasta or .fast. It won't take that long. The longest amount of time it's going to take is to just read through the blast output file. It doesn't take all that long. We're done. And so we can at least look through this reference orf output. So we can list that real quick. Or sorry, reference. Right. So now we have the Trinity, we have the transcript name, plus this Q number, and a CDK number, or the gene name. And so I'm gonna split my screen for a second so we can kind of see how both look. Because I've modified the script to only provide information that I only care about, right? So I'm going to grep this number, this Q number, out of the blast output file. I'm going to do this in a new window. No, I won't. I'm just going to pipe it to head. Okay. All right. So this is how it appears in the blast output file, but I've amended it so I get rid of human. There is a reason for this, because I want to see the number of unique genes. So what's going to happen some of the time, the best hit won't be from a human gene. It's just a product of blast. It's a property of blast. It's a property of working with transcriptomes, because whenever we work with a transcriptome with simply, we're not actually working with exact sequences of what we would find in the genome, right? It's often called a gene model. It's very, very close, but it's not perfect, and we can't right now validate that it is indeed perfect. But it is certainly better than nothing, and it's pretty close. So some of the time, we will return, the best hit won't be from your species of interest, right? Even though I've run this on human, there are human reference genes in here, because this simply isn't perfect, because sometimes we get short sequences, things like that. Sometimes the best hit will be a mouse gene, or it will be from a dog, or from a rat, something else other than human. And so I don't really care about where that gene annotation came from, what species that directly came from. All I really care about is the gene name. Nonetheless, I do hang on to the Q number, because that Q number is going to be directly related to CDK20 in human. So I do have both. I maintain both by hanging on to the Q value, this Q number, the specific accession number, and the gene name. It's just whenever I want to search for the number of unique genes, if I grep out CDK20, I'll most likely see both mouse and human genes from that. Right, let's see if I do that. CDK20. Right, so like everything done, right? That's a CDK20. This is a blast rumor. This is a blast output file. This is from Danny O'Rerio. That's a zebrafish. Right, there's rat. And so I just don't want these species annotations. If it comes up with a gene name, it's probably that gene. It's just I don't really care what the species source is. It's close enough. The homologs was the best. The homologs are present. Anyways, get rid of that. And so right. And so all this is period delimited. Like it's easier for me to work downstream, so I kept this as period delimited. Right, transcript name PL, just because it's there, period, and then the protein annotation. And again, so ultimately, I'm going to map reads back to this. We're going to use this file. We're going to keep using this file in the next step that I'll talk about on Friday. Is actually mapping reads, our clean reads, back to this reference. And so all this stuff is present. And here, and here's a nice example. Here's kind of a nice example between these two genes. So look, in these two, we have the same annotation. CDK20, CDK20. But this P1 and P2 effectively denotes these are separate alternatively spliced transcripts. at least says Trinity. This is probably a partial, but either way, both of these transcripts have this exact same annotation. They probably have the same gene source. And so we're going to see that pretty commonly in this output file, but I'm not going to touch it. I'm going to leave it alone, because what we might be seeing is this might be a partial gene. This might be partial on the 3' end. Or right, so just because there's an ATG doesn't necessarily mean that's the right start codon. And so it's very possible this is like the 5' region of that gene, and this is the 3' region of that gene. Or an alternatively spliced transcript, something along those lines. So I'm hanging on to everything. If it was annotated, I'm hanging on to it, and I'm not going to touch it. RNF, but yeah, so all of these transcripts are now annotated to the best of our very quick ability. Okay, and so again, let's kind of explore some of the stuff that's in this file. At least let's explore the number of annotations. So how many unique genes were actually annotated here? So I'm going to grep some stuff out, grep and cut some stuff. I'm going to grep the header. Then I'm going to cut on this, on the pipe. I'm going to cut that character. And so all I'm left with is the actual gene name. And so I'm going to cut with the delimiter. I'm going to use that as a pipe, put that in quotes. Otherwise, you'll read it as a pipe and it won't work. It has to be in quotes. And -F2, because if we delimited this is all going to be one column, and that's going to be the second column. So we're going to delimit on the second field. Make sure that works. Yes, that works. Okay, so now we can see all the unique annotations in this. Furthermore, this is unsorted, so I'm going to sort it out. And I'm going to run and so what you'll see if I sort this is we'll see the same annotation pop up over and over and over again. Some annotations will only be present once, but others will be annotated multiple times. Again, the imperfection of assembling and the craziness of biology, the imperfection of biology, alternative splicing, etc., etc. We'll see multiple annotations from many a gene. All right, and I just keep on going through. Again, some are annotated only once. Others are annotated more than once. And so we can kind of see how many genes were actually annotated more than once. So I'm going to do a sort. I'm going to do a unique and a C. Unique dash C, so I can count the number of unique annotations. Let me list that real quick. Okay. Right, so that transcript, that gene was present in 7 transcripts. That's an 11, 12, 2, etc., etc., etc. And so I'm going to sort this one more time. I'm going to sort on in. I'm going to sort dash in. To basically sort out on this on the count number. So if a G, let's just see what happens. Okay, I'm going to go to the very end. So this is the very end of that sort unique. And so the most commonly annotated gene here was this LORF2. Don't know what that gene is, but it was annotated in 144 different transcripts. Now the thing is, we see a lot of these genes annotated more than once. But if we scroll up, I'm just scrolling up in this, they reduce kind of quickly. And I want to say I expect, and you should almost expect, somewhere between 5 and 10 alternatively spliced transcripts in a transcriptome for every gene. That's kind of how biology is, without how crazy alternatively spliced transcripts get. Furthermore, again, just imperfection of assemblers. Their genes can be fragmented when they're assembled. And so like we may have a 5' region, we have a 3' region, but we're missing the internal stuff. So we're going to leave it all there. We're just going to leave it all there. Now if I scroll to the top, there should be something to notice is that most of these annotations are present only once. The solid majority of them. If I plotted this out in the form of a histogram, most genes are annotated only once. Some are annotated twice, but the more annotations that are present, the fewer times this happens. Anyway, so let's just do a quick count on all of this to find that among these 78,000 transcripts present, 14,000 unique genes were actually annotated, right? So that's how many raw genes or unique genes are actually here, 14,000. And that's consistent with what I typically see in a mammalian transcriptome, somewhere between 8 to 14,000, usually not more than 15, but it probably comes out to an average of about 10,000. And we also have to recognize this as testis. Testis is doing all kinds of crazy things. It's going through mitosis at the same time it's going through meiosis. Its sperms are maturing. Sperm do a lot of weird things. But one thing I'm going to grep out a specific gene. I'm going to grep TERT. And so that should be here. So TERT is a telomerase reverse transcriptase. So it's the main telomerase gene. And since this is a somatic, or this is not a somatic, it's a germline tissue, you expect telomerase to be expressed. And so telomerase was here, it's expressed, it was assembled, and annotated. So that at least makes biological sense. And so we should see a lot of other testis-specific genes. I'm going to try one more. I'm a little hesitant. It could come back to bite me. I'm going to grep on a peewee, which should also be a testis gene. And that did not come back out. So that's a little nervous. That makes me a little nervous. But peewees are a long gene. Whatever. It may not even be annotated as peewee. Honestly, in the Swiss it may not be annotated as peewee. I'm looking for a word that doesn't actually exist. So who knows? I know TERT for sure is real. Anyway, now just one more time. I hate testing stuff. Let me try iwi. Nope, I got nothing iwi. Anyways, moving on. Okay. So this is what's going to be our reference and downstream analyses. Right, again, if we don't have a reference CDS library, this is probably the easiest and quickest way to make it from FASTQ RNA-seq data. Assemble it, find your open reading frames, blast your open reading frames to the SwissProt, and add the best hit to the ORF. If there is no hit, leave it alone. If there is an ORF and it's annotated, use the best. But even still, this will probably be 98% accurate. And it's just a property about the messiness of vertebrate genomes and paralogs. Because sometimes if a transcript is too short, if it's too kind of murky, a paralog will be improperly annotated. So you may be looking at, your gene may actually be like CDX3, but it'll be annotated as CDX1. Again, just because BLAST isn't perfect, nothing in biology is perfect. Biology is inherently messy. A lot of problems can't be entirely solved, right? They're just solutions. We have solutions, but perfection is almost impossible in biology. We work with what we have. We do what we can with what we have. And we run experiments to make sure that what we're doing is proper. And so again, like what's going to happen now, how we're going to analyze gene expression is going to be a little unique. It's not normally how I do it. But if there's still time left in the semester, because this is kind of the last project I have for the semester, we're gonna do this. We're gonna do differential gene expression stuff, and see what comes out different in our liver tissues. But if there's enough time, I'm going to try to do it a different way. Something that I would consider to be a little bit more proper, because we do kind of have a reference. So we're dealing with turtles. There is something that a closely related species to our turtle of interest on Ensemble that has reference CDSs. So if you have a reference, it's better to use the reference. But since I'm also kind of just playing around this class, doing my own little experiment, seeing what happens, we're going to use our assembled and annotated reference first, just to see what happens, and then compare that to, if we have time, we'll compare that to mapping reads to a real reference, like a much more curated and robust reference set of genes. All right, so that's all I'm going to do for today. On Friday, I will do the mapping, so we'll go through the mapping step of finding out where our clean reads go to each transcript and how many transcripts have reads mapping to them. All right, so I'm going to call it there, and I'll see you all again on Friday. So again, just get poll to get the annotate ORFs, the FASTA file, the BLAST file, save that. Should be enough. That's all. That's kind of the last steps. All right, I'll see y'all later.